In this video, we're going to focus on a specific category of proteins called enzymes. Enzymes are proteins involved in catalyzing reactions. Chemical reactions that convert substrates into products are essential for all sorts of biological processes, including metabolism, without which life could not occur. Enzymes help increase the speed of these reactions so that they can occur fast enough to sustain life. They are catalysts, which means that they increase the speed of the reaction, but they're not actually used up during the reaction like a substrate would be. So a single enzyme can catalyze multiple reactions. Let's take a look at this sample chemical reaction here. Here we have substrate A and substrate B coming together in order to form product C. The first thing that needs to happen for this reaction to occur is substrate A has to be in close proximity with substrate B. So let's say that this reaction is happening in the cytoplasm of a cell. So here we have our cell, and we have its nucleus, maybe some organelles here, and then we've got some substrate A, some substrate B, and then also a ton of other molecules that are not involved in this reaction. So when you think about what the environment looks like for substrate A and substrate B, the likelihood that they're going to bump into each other by random chance alone is fairly small because there's all this other stuff in the cell that the proteins can bump into. This is especially going to be the case if the substrates aren't found in the cell at a high concentration. But let's say that these substrates do find one another in the cell. The next thing that needs to happen for the product to form is substrate A and substrate B need to form bonds with one another. But remember that substrate A and substrate B have already formed stable intramolecular bonds between their own atoms. So in order to begin forming bonds between one another, some of the bonds in substrate A will have to become less stable, and some of the bonds in substrate B will have to become less stable in order to form new stable bonds between substrates A and B. This new unstable state is known as the transition state. It occurs somewhere between the point where substrate A and substrate B are stable on their own, and the final product C is stable. These two substrates must reconfigure into this unstable transition state prior to forming the product, and this process requires energy. The minimum energy required for this reaction to occur is called the activation energy of the reaction. We can look down at this graph here to follow the energetic requirements of the reaction without an enzyme present. So here on the y-axis, we have the energy required for the reaction to proceed. And here we have the reaction coordinate, which is basically just the time that the reaction is occurring. At the beginning of the reaction, the substrates start at a certain level of energy, determined by the stability of their intramolecular bonds. Then, as they collide and start to form the transitional states, those intramolecular bonds are going to become less stable, and this process is going to require more energy to occur. Eventually, the transition state reaches a peak level of instability, and gradually new bonds begin to form between substrates A and B to form the product. And as this occurs, the energy drops back down to the final energy of the product. Notice that the energy level of the final product is lower than the energy level of the initial substrates. This means that this reaction is energetically favorable, and if given enough time for substrates A and B to collide with one another with enough energy to overcome this activation energy of the transition state, this reaction could proceed. But for some biologically relevant reactions, the time that it would take for this to happen is way more than the lifespan of a human. This activation energy is a big barrier to reactions proceeding at a reasonable rate. What enzymes do is they lower the activation energy of the reaction, and they can do this through a few different ways. One way is that the enzyme, which remember is just a protein with its own primary, secondary, and tertiary structure, can be folded in such a way that it creates binding sites for the substrates. So for example, looking at this green enzyme here, there is one binding site for substrate A and one binding site for substrate B. These binding sites are shaped in a way that they can interact with specific molecules and bring those molecules close together in order to facilitate the transitional state forming. One way to conceptualize this is to think of the substrates as tennis balls rolling across the floor. So let's say that this is substrate A and this is substrate B. What we need for product C to form is these substrates need to collide with one another at a high enough rate that it overcomes that activation energy. However, when you think about what happens when two tennis balls collide at a fast rate, they're just going to bounce off of each other, right? So rolling a tennis ball across a flat floor is analogous to the situation where there's no enzyme. Even if these tennis balls manage to come in close proximity with one another, they're unlikely to be there for long enough to actually form the transitional state and the product. Now let's think about what would happen when we do have an enzyme. This would be analogous to rolling a tennis ball across a floor that has a ditch shaped for two tennis balls. Now when you roll these tennis balls across the floor, two of them might get caught in this little pocket. Now these tennis balls can have a prolonged interaction, and that's basically how enzymes work, by forming a little pocket for the substrates involved in the reaction. When the substrates bind to the enzyme binding site, they can also subtly change shape in a way that makes the transitional state more likely to occur. Many enzymes also stabilize the transition state itself by providing a source for electrostatic interactions that stabilize the molecular bonds as they shift from the substrates to the product. By bringing the substrates closer together and stabilizing the transitional states, enzymes lower the activation energy that's required for the reaction to proceed. This means that the reaction can proceed much more readily. And then once the reaction occurs, the product unbinds from the enzyme, and the enzyme is free to catalyze another reaction with new substrate molecules. Enzymes can be classified by the kind of reaction they help carry out. Oxidoreductases transfer electrons from one molecule to another. An example of this is pyruvate dehydrogenase, which helps convert pyruvate to acetyl-CoA immediately prior to the citric acid cycle. Transferases are responsible for transferring a chemical group to a molecule. For example, methyltransferase transfers a methyl group to a histone, which causes DNA to be wrapped more tightly and be less accessible for transcription. Hydrolases cleave an intramolecular bond by using water. For example, lipases break down fats into fatty acids and diacylglycerol by transferring atoms from water to the molecule and causing an intramolecular bond to break. Lyases, on the other hand, cleave a bond without using water. An example of a lyase is adenylyl cyclase, which breaks ATP down into the key signaling molecule cyclic AMP. Isomerases convert a molecule from one isomer to another, such as phosphoglycerate mutase, which moves a phosphate group from the third carbon on glycerate to the second carbon as part of glycolysis. Ligases help form bonds. For example, DNA ligase helps DNA transform phosphodiester bonds and is important for both DNA repair and replication. And finally, translocases are enzymes that are responsible for transferring molecules across the plasma membrane, such as ornithine translocase, which transports ornithine from the cytosol to the mitochondria as part of the urea cycle. You'll notice that all of these enzymes have a name that ends with ase. And in fact, enzymes are often named after the substrate that they are acting upon with the suffix ase added on. So for example, lipase is an enzyme that acts on lipids. The key point to remember from this video is that enzymes are a type of protein that catalyze chemical reactions by lowering the activation energy. Enzymes are involved in all sorts of important biological processes, which makes them quite useful for diagnosing and treating disease.